Hi everyone. My great uncle taught me three things I'll never forget: how to ride a bike, how to do geometry, and how to tell a story. However, in his final years, as Alzheimer's slowly took control over his life, he forgot the very lessons he once passed down to me. Alzheimer's is a neurodegenerative disorder that slowly erodes memory and cognitive function. At the Manuki lab, neuropathologists noticed something interesting. A rare type of cells, a rare type of cell in the brain's choroid plexus epithelial tissue called oncocytes. For context, the choroid plexus is a region of the brain that produces cerebrospinal fluid, which is fluid that cushions and protects the brain against impact. Oncocytes are a special type of cell that are characterized by an abundance of mitochondria, the powerhouse of the cell. While they're uncommon, researchers at the Manuki lab hypothesize that they may be linked with the progression of Alzheimer's. However, right now the issue with studying these type of cells is that they're so incredibly rare, and underneath the microscope, they look almost identical to healthy cells. This image on the right really shows that, where the oncocytic cells are labeled in red and the healthy cells are unlabeled. As a result, pathologists have to scan slides one cell at a time, which is slow, exhausting, and prone to many errors. To fix this, I look towards using AI. Specifically, I adapted a deep learning model called HoverNet to automatically find and flag oncocytes and whole slide images of choroid plexus tissue. The idea is to train a model on tissue samples where pathologists have already labeled each oncocyte. From this, the model learns how to separate every cell's nucleus, classified as oncocytic or not, and does this um, at thousands of cells at a time. So far, the early results are very promising. The HoverNet model is able to detect various different cell types and different tissue types, and does this with a very high accuracy that matches what pathologists are able to do, but at a much greater speed. Next steps in my research project include fine-tuning the model specifically on the annotated and labeled choroid plexus epithelial tissue, and then testing it across different staining protocols. The ultimate goal with this project is to make a freely available and open source tool for clinicians and researchers to use worldwide. Also, something that I really want to importantly note is that this technology isn't intended to replace pathologists, but it's there to give them sharper tools. AI can take care of the tedious manual counting steps so that pathologists can focus on diagnosing and treating diseases. For me, this work is very personal. Alzheimer's stole memories from my great uncle, and I know so many families who have lived similar stories. Through this project, I hope to turn that loss into something meaningful and create a tool that helps preserve memories, protects families, and gives hope against fighting against Alzheimer's. Thank you for your time, and here are my acknowledgments.